Now it is my pleasure to introduce you to your speakers for today's webinar. And first, I'd like to introduce you to Dr. Amanda Finan, and she's a head of R&D IHC with Cerba Research. Amanda joined the R&D team focused on IHC at Cerba Research in 2018, and at the beginning of 2020 was promoted to manager of IHC R&D. Amanda has extensive experience in physiology with a particular focus in immunohistochemistry and imaging. And next, I'd like to introduce you to your second speaker, and that is Dr. Aten Akirav, and he is the VP of Business Development, Global Lead of Biomarkers at Cerba Research, where the biomarker team is focused on providing a comprehensive solution for preclinical and clinical research. He is also an assistant professor at Stony Brook University in New York. He has authored more than 25 peer-reviewed papers and book chapters. Now it is my pleasure to pass over the controls and mic to Aten. So Aten, when you're ready, you may begin.
Thank you so much, Sonia, for the kind introduction. First off, I wanted to thank Xtalks for hosting the call, and also extend my special thanks for the audience. I know you're busy, and we really appreciate the fact that you took an hour of your time to join us today. So just as a quick agenda on what we plan on covering, Amanda and I will be discussing the general landscape of biomarkers in oncology, what it means, how it's being approached by the market, as well as Cerba. We will then take a deep dive into in situ biomarkers, looking at the application of IHC, moving into the power of multiplex IHC approach, which allows scientists as well as end user look at multiple biomarkers on a single slide, and finally give you kind of a quick review of how Cerba Research looks at biomarker development in IHC, and what's the most common pathway that we take when we bring a biomarker to a study and finally to the market. So I figured it would be a good idea to give you a quick rundown on who is Cerba Research and what do we do. So Cerba Research has been around for nearly 4 decades. We are one of the most established CRO central laboratories currently supporting clinical trials. We are divided into 4 main segments. The first segment has to do with our bread and butter, which is clinical trial laboratory services. This service has been around since 1983, and it really provides soup to nuts service for clinical trials. Anything that has to do from sample management, sample testing, logistics, database setup, and reporting of in data integrity into the FDA and other regulatory agencies. In addition to that, we have a dedicated arm that deals mainly with in vitro diagnostics, so IVD. This arm is experienced in bringing new assays. As you can imagine, we've been extremely busy with the COVID situation. What we do there is we have access to multiple types of platforms and predicate assays, which are quite important when submitting for a 510K or a PMA application. And finally, the last 2 segments, the translational research segment and the biomarker and CDA segment, which Amanda and I are part of, really focuses on the biomarker aspect of both preclinical, retrospective samples, and clinical prospective clinical trials. This effort is geared towards bringing both off-the-shelf biomarker development, as well as bringing brand new, previously untested biomarkers to any type of preclinical and clinical research. So the type of methods that we actually employ in the translational research arm of Cerba really encompasses all of the common methods that have been used by researchers and sponsors in clinical trial and preclinical trial. These include PK/PD capabilities, flow cytometry, and IHC for cell phenotyping, genetics and genomics using NGS, which I will touch upon during the call, as well as molecular biology and metabolomics. And the beauty of this proposition is that everything is really under one roof or under one umbrella, if you will, which makes the ability to connect data, interpret it, and report it back quite easy and seamless. We are truly global. We have presence in all major continents. These include not just logistics support. In many cases, we also have wet lab capabilities. So that means that in clinical trials or samples that have to travel from one part of the world to the other, we can offer a comprehensive solution on all multiple aspects. I would like to highlight the fact that because we have access to wet labs and because we have a battery of dozens of in-house PhDs at CERBA, we are able to provide SOP transfer between the different sites. And that's extremely important because when one looks at bringing the same assay in different location, data integrity, the type of platform that is being used, as well as the data interpretation, all ensure that you as a scientist or as a sponsor have an apple to apple rather than an apple to oranges type of capability. All right, so I will jump right into the topic at hand, which is looking at biomarkers in oncology with a kind of a strong focus on IO or Immune-Oncology. And the first slide is kind of a sobering testament to the fact that the probability of success in clinical trials is currently quite low. We all know that oncology trials are the most common form of clinical trials today, but unfortunately, they do present with the lowest success rate out of all the therapeutic areas. In this report by Harrison et al. From 2016, they've highlighted the fact that this trend has actually remained quite steady since 2007, which really suggests that we must rethink the way that clinical trials are being designed and ran. And finally, if you look at the main cause for failure to bring a new drug, compound, biologics, it doesn't matter what is being studied from phase 2 to phase 3 as well as to approval, it is clinical efficacy or the measurement of clinical efficacy that is the major cause for failure. So in a subsequent paper published by Wong et al. Last year in 2019, they looked at the probability of success with a focus on oncology, metabolic, and CNS trials. And what you can tell from this graph is that out of all the clinical trials, whether it's phase 1, phase 2, or phase 3 or approval, phase 1 has the highest success of rate, and that's not surprising because the bar for phase 1 is relatively low. There's normally no focus on efficacy, and mostly the measures focus on safety. However, the success rate does drop and does not improve as one moves throughout the clinical trial development. So you can see that the blue bar, the dark blue bar for oncology actually is the least performing out of all the three fields of clinical trials depicted here. And finally, only about 3.4% of all oncology trials actually make it to approval. And this is kind of a sad story of only 1 in 20 of all clinical trials being successful at a best case scenario. So there has to be a way, or we should think of a way of enhancing this probability of success moving forward. So the beauty of this report by this group is that they actually segmented out those trials that implemented biomarkers. And when they did that, they actually defined biomarkers as factors that are being used to select patients, enhance safety, and serve as surrogate clinical endpoints. One of the interesting parts that came out of this report is that out of the 10,000 clinical trials or data points, I should say, that were analyzed in this case, only about 10% or 1,100 of those actually employed biomarkers. And this is quite shocking, to be honest, because it means that 90% of clinical trials that are out there have not included biomarkers for their, as part of their clinical design. However, those that did decide to include biomarkers benefited, and also in big time, because the rate of success actually increases approximately 5-fold by the inclusion of biomarkers in the clinical trial design. So this is a nice indication that undertaking the sometimes labor-intensive, cost-intensive has for bringing biomarkers can pay off at the end of the trial. Okay, so what is a biomarker? And I chose to take the definition as it's posted by the Food and Drug Administration, the FDA. Ultimately, this would be the one of the regulatory organizations that will look at the results, and they define biomarkers as a characteristic that can be measured as an indicator of normal pathogenic processes in response to intervention, therapeutic intervention, as well as medical devices. What's interesting in this definition is that you can see that the type of biomarkers listed on the slide would probably now fall under the standard of care, or I would call very established biomarkers in the market today. Blood glucose has been around more than 100 years for the measurement and treatment of diabetes. Tumor size through imaging is also being commonly used in both standard of care as well as clinical trials, and blood pressure is, of course, a standard of care biomarker that are being used. But I think most of the scientists, and I'm sure the ones in the audience today, would appreciate the fact that biomarkers today, the term biomarker, is a lot broader. It's in fact a big umbrella that encompasses several different aspects of measuring biological responses. All right, so when we look at cancer biomarkers, we can actually divide those roughly into biomarkers that can be viewed locally or systemically. When looking at systemic biomarkers, they provide us information on tumor macroenvironment. In other words, the factors that change in the body as a whole. We can use several different types of bodily fluids in order to measure those biomarkers. Those could be plasma, serum, saliva, and urine. And in addition to that, an up-and-coming field today actually looks at circulating cells in cancer, looking at tumor cancer, circulating tumor origin, also known as liquid biopsies, has been quite a powerful tool in order to measure and assess tumor state, as well as tumor phenotype and genotype, looking at the immune cells that are circulating to look at the level of immune activation to the tumor, again, very relevant for immune-oncology. And finally, looking at healthy cells, or I would call it the stroma, to see whether changes in non-cancerous cells can actually also provide biomarker information. Local biomarkers are quite different. They look directly at the tumor microenvironment. Those are especially relevant for solid tumors. They can give us information on the tumor structure. They give us information on the tumor origin. One can look at metastasis and see where was the original tumor coming from. There's an ability to look at the stroma, how the stroma actually responds to tumor, for example, the generation of vasculature in order to support tumor growth. And finally, again, with high relevance for IO, is to look at the immune phenotype, or the type of the immune cells that actually make it into the tumor and take or do not take the action to eradicate the tumor. What's evident, whether you're looking at systemic biomarkers or looking at local biomarkers, is that the techniques that are used in these two types of biomarkers are actually shared. Here's kind of a really not a very comprehensive list of techniques that are being used today, but just to give you a flavor that whether you're looking at cell-based, molecular, genetic, and so on, the tools are out there. Many of them are, of course, available in CRO organization such as CERBA. So the first focus, since this is an IO-based focus talk, is to look at circulating lymphocytes in cancer. We all know how important the immune system is in protecting us from viruses, bacteria, and fungi, but we also know that the immune system plays a very important role in immune surveillance, looking for cells that undergo transformation, become cancerous, and in most cases is actually quite successful in eradicating those cells and keeping cancer in check. However, in those cases where cancer does manage to escape immune surveillance, when we look at circulating lymphocytes, we can get several key pieces of information by using biomarkers for these cell types. It gives us a snapshot of the immune cell types that are in the blood. It also gives us a measurement on how well the immune system is activated, whether the immune system has become allergic or tolerant to the tumor, and whether we were able to introduce or induce a memory response, such as in the case of using immune checkpoint therapy or engineered CAR T cells. Changes in activation over time are also important. This is especially important for prognosis, identification of new therapeutic targets by identifying suppressor or activating molecules that are being upregulated on the immune system. We can then use a combination therapy in order to wake up the immune system to attack the cancer, just as an example. And finally, and equally important, is the use of immune phenotyping for patient stratification. Again, extremely important in IO to make sure that the right patient gets the right type of drug or experimental drug during the clinical trial. The type of cells that we can phenotype using circulating lymphocytes is really wide. We can look at the subsets of immune cells. We can look at the immune cell activation. In the case of blood cancers, we can actually stain the leukemia cells and assess the level of activation and the phenotype as well as genotype. And finally, we can use circulating factors in order to do pharmacodynamics as well as receptor occupancy. So jumping into the methods that are being used to characterize circulating lymphocytes, one of the most well-established and older methods that have been out there is, of course, flow cytometry. Flow cytometry allows the tagging of any type of cell, in this case, in this example, immune cells, with multiple types of antibodies. In fact, today, FACS instruments can provide more than 20 different biomarkers per cell, an extremely powerful tool to get a full characterization of a single cell. In this slide, I'm showing you a relatively straightforward and simple analysis of memory T cells. This is data coming directly from Cerebral Laboratories, from our flow cytometry team. In this case, there are cells that have been purified from plasma. They then get gated on live cells, subsets of different immune cells. The focus here are T cells. You can see CD4 helper T cells and CD8 cytotoxic T cells being further gated with a series of antibodies, and then looking at central and peripheral memory T cells, again using antibodies against key T cell receptors, chemokine receptors, and so on. And as I mentioned, this is a single example of a really powerful tool that has been used in both preclinical and clinical research. But looking at circulating cells got a significant boost with the introduction of mass sequencing through next generation sequencing. So next generation sequencing, as described in this manuscript by Shea et al. Earlier this year, used NGS to look at the repertoire of T cell receptors in engrafted T cells in patients receiving CAR T cells. I mean, this is absolutely phenomenal. I mean, looking at cells that were engrafted in humans was probably science fiction 5 or 10 years ago. And today we have the ability to engineer cells, put them back into the patient and look at their evolution. In this case, you see a panel of patients with ALL. Each one of these graphs represent a single patient, and on the bottom, I'm sorry, you can see non-Hodgkin's lymphoma patients. Again, about 5 patients here. Each one of these dots represent a single clone that was identified prior to engraftment into the patient. And what you can see here is the kinetic of the expansion of the clone over a 30-day period of time. Again, a really remarkable and powerful methods to assess the response to the tumors. But looking at T cell receptor is not the only aspect of using NGS. The same group also used NGS in order to look at the expression of several different genes produced by T cells. Why is that important? introducing a T cell into a cancer patient has to elicit a favorable response, right? That would actually cause the tumor to shrink and eventually, hopefully, disappear altogether. Here they used a gene array, an NGS approach, looking at the early and late stages post-engraftment, and they've identified several different genes that pertain to chemokine, cytokine, as well as other cytotoxic and growth factor produced by T cells. I wanted to give you a single gene that they actually focused on here. As some of you may know, the robust expansion of CAR T cells post-engraftment is extremely important to elicit a good response. But the flip side of it, or the other side of the coin, is that we don't want these engineered cells to kind of take over the body and grow uncontrollably. What they have done, they've used NGS to look at the biomarker of T cell proliferation, also known as Ki67. In fact, it's a pan cell proliferation. And what you can see is that in all of these 4 cases of CLL and non-Hodgkin's lymphoma, the level of proliferation was indeed reduced late post-engraftment. Again, something that was really unheard of just a decade ago. So immune cells, as they circulate in the system, are not just passive factors helping to destroy tumor cells upon contact. They are also active secretors of soluble factors that can either stimulate the destruction of the tumor, but unfortunately, in some cases, can also inhibit the immune system from attacking and destroying the cancer cells. And therefore, we should have a way to measure the ability of these cells to secrete specific factors that are relevant for the cancer. We can look at secretion of factors at cells that infiltrate the tumors. Those are known as TILs, and I'll talk about them in just a couple of minutes. But we can also look at the total level of secreted factors. In this case, it could be chemokines or cytokines systemically. And it's that crosstalk between the systemic response and the tumor response that seems to be most important in terms of facilitating tumor destruction and shrinkage. So how can we measure these secreted soluble factors? And again, 2 very robust approaches that we employ at CERBA routinely. One of them is to look at direct secretion from the cells. In this case, cells are purified. It could be either directly from the tumor, but in this example, they are purified from the circulation. One method, which is well established, is known as ELISpot. In this method, T cells, and sometimes B cells, are exposed to a specific antigen. The antigen stimulates the cells to secrete all the goodies, if you will, that it produces in order to destroy the tumor. And this can then be enumerated using a colorimetric assay. In addition to that, again, using our extensive flow cytometry capabilities, we're able to do intracellular cytokine staining. T cells, in this case, cytotoxic CD8 T cells, are stimulated with an antigen, and then looking at the production of 2 highly pro-inflammatory factor, interferon gamma and TNF alpha, produced within the cell. Again, a very powerful tool that is a derivative of flow cytometry that I showed you before. Another approach is to look at soluble factors that are present in the media or in the blood. Whether you work with fractionated blood, serum, or plasma, or you're looking at stimulated cells in vitro or ex vivo using, in this case, a 96-well plate, we are able to take that material and then test for the presence of specific cytokine, chemokines, antibodies, you name it. In the old days, the technique that used to come to mind is ELISA. We have now adopted at CERBA the MSD platform, which is allowing us to multiplex several different types of cytokines and chemokines in a single well. The way that it works is through electrochemiluminescence. Each one of these little electrodes will measure the presence of a specific T cell or B cell secreted factor that can then just be enumerated. This is an extremely powerful technique, not just because of the cost saving compared to multiple ELISAs, but also in the case where serum samples are precious, such as in the case of pediatric oncology. And I will close my part of the presentation by kind of giving a segue to Amanda's segment, which is what happens when we're zooming into the tumor microenvironment. And a big focus on IO is on tumor infiltrating lymphocytes, or TILs. And TILs are technically those cells that you see circulating in the periphery, homing into this tumor and exerting either a positive or a negative effect on the tumor. Some of the cells that we focus on are of the innate immune system, such as dendritic cells, macrophages, and myeloid-derived suppressor cells, and NK cells, but we also are able to look at cells of the adaptive immune system, such as regulatory T cells, effector T cells, and helper T cells, and so on. And by combining the systemic view of the circulating lymphocytes together with looking at the tumor, one is really able to get a complete picture of the immune activation, especially when focusing on an IO trial. So, Sonia, I will hand it to you to present the poll question for our audience.
Yes, I will do that right now, Eitan. I'm going to launch our first poll.